Hello students, I am teacher Svetlana and in our today's lecture we will learn about urine formation and concentration of urine, which is covered in the chapter Excretion and Osmoregulation. So let's begin. Urine formation. The process of urine formation is completed in three successive steps as ultrafiltration, also known as glomerular filtration, selective reabsorption, and tubular secretion, also known as argumentations. So let's see each of these steps in detail. Ultrafiltration or glomerular filtration, the diameter of the efferent arteriole is greater than the efferent arteriole. We are already aware of this fact now. Here you can see here the efferent arteriole has a greater diameter as compared to the efferent arteriole. Diameter of the capillaries is still smaller than both the arterioles. Due to such difference in diameter, blood flows with great pressure through the glomerulus. So here inside the blood flows with great pressure. This is called as glomerular hydrostatic pressure, GHP, and normally it is about 55 mm Hg. This pressure is opposed by osmotic pressure of the blood, normally about 30 mm Hg, and capsular pressure, normally about 15 mm Hg. And hence the net effective pressure, EFP, is 10 mm Hg. So here the effective pressure worked out after the differences in the pressures is 10 mm Hg, which is considered as the effective filtration pressure. Furthermore, students, walls of the capillaries are extremely thin. Under the effect of high pressure, walls become permeable to major components of blood. That is, they allow major components of the blood to pass through them, except blood cells and macromolecules like proteins. Thus, plasma, except proteins, ooze out through the wall of the capillaries. And about 600 ml blood passes through each kidney per minute. The blood plasma flowing through the kidney, that is the glomeruli, is filtered as glomerular filtrate at the rate of 125 ml per minute. So here you have the filtrate that comes out is called as the glomerular filtrate. Glomerular filtrate, also called as deproteinized plasma, that is plasma apart from or except the proteins, and we have the primary urine, which is alkaline. It contains urea, ammonia acids, glucose, pigments, and inorganic ions. Glomerular filtrate passes through the filtration slits into the capsular space and then reaches the proximal convoluted tubule. So here have a look again. You have these slits here, and the filtrate passes through the slit and reaches the space which is called as the glomerular capsule, and then it reaches the PCT, that is the proximal convoluted tubule. The next step is selective reabsorption. The PCT is a place for reabsorption. Have a look at the diagram here. This place is where the reabsorption is taking place. It is highly coiled so that the glomerular filtrate passes through it very slowly. Columnar cells of the PCT are provided with microvilli, due to which absorptive area increases enormously. This makes the process of reabsorption very effective. So it has the columnar cells have special microvilli which increases the surface area, and hence the process of absorption also increases to a very great extent. These cells perform active, ATP mediated, and passive simple diffusion reabsorption. So both active and passive absorption is taking place. Substances with considerable importance, that is high threshold, like glucose, amino acids, vitamin C, calcium, potassium, sodium, chloride ions are absorbed actively against concentration gradient, and low threshold substances like water, sulfates, nitrates, etc. are absorbed passively. In this way, about 99% of the glomerular filtrate is reabsorbed in the PCT and also in the DCT, that is the distal convoluted tubule. Tubular secretion or argumentation. So finally, the filtrate reaches the distal convoluted tubule via the loop of Helm. Have a look at this diagram here. It shows at which stage what is reabsorbed from the filtrate. So here we were first at the PCT, and here now we are at the DCT, and here you can see here that the filtrate has passed through the Helm's loop. Peritubular capillaries surround the DCT. Cells of the distal convoluted tubule and collecting tubule actively absorb the wastes like creatinine and ions like K plus, H plus from the peritubular capillaries and secrete into the lumen of the DCT and the CT. So here you have the secretions that are secreted here in the lumen of your distal convoluted tubule and the CT, which is the collecting duct or the collecting tubule, thereby augmenting the concentration of urine and changing its pH from alkaline to acidic. Secretion of H plus ions in the distal convoluted tubule and the collecting tubule is an important homeostatic mechanism for pH regulation of the blood. This process is called as tubular secretion or augmentation. So here you can see that we have secretions of the potassium and H plus ions, and these secretions collectively help in maintaining the pH of the blood, and this process is called as tubular secretion or augmentation. Tubular secretion is the only mode of excretion in marine bony fishes and desert amphibians. Now students, we will have a look at the concentration of urine. Now under the conditions like low water intake or high water loss due to sweating, humans can produce concentrated urine. It can be almost 4 times concentrated, that is 1200 milliosmol per liter, than the blood, that is 300 milliosmoles per liter. Now students, just for your information, the unit of osmotic concentration is called as osmol. Milliosmol, which is denoted as MOSM, is 1 part of 1000 part of an osmol. Now for this purpose, a mechanism called as countercurrent mechanism is operated in the human kidneys. So there is a mechanism to concentrate our urine in the human kidneys. We will have a look at this mechanism. Students, the lymphs of the Helen's loop of the juxtamedullary nephrons and the vasa erecta operate countercurrent mechanism as follows. So this is your nephron, which is of the juxtamedullary type of nephron, and this is the vasa recta. If you can recall, this vasa recta is basically the capillaries which surround the Helen's loop. This mechanism involves the passage of fluid from descending to ascending limb of Helen's loop. Flow of tubular fluid is in opposite direction through both the limbs, hence the name countercurrent or opposite flow. In case of vasa erecta, blood flows from the ascending to the descending part of itself. Wall of the descending limb is thin and permeable to water, whereas that of the ascending limb is thick and impermeable to water. So here you have your descending limb which is permeable to water and the ascending limb which is not permeable to water. It is thick and it is not permeable to water. You can see here in the diagram water is being secreted, but here no water is being secreted as water cannot pass through this lymph, the ascending lymph, as it is impermeable. In the region of the descending lymph, water diffuses from the tubular fluid into the tissue fluid, due to which tubular fluids become concentrated. Ascending lymph of the Helen's loop is thick-walled and its cell can reabsorb only sodium and chloride ions from the tubular fluid and release it into the tissue. So here only your sodium and chloride ions can be absorbed and released into the tissue. Due to this, the tissue fluid around the descending limb becomes concentrated. This makes the more water to move out from the descending lymph into the tissue fluid by osmosis. So this makes here, because here the fluid becomes concentrated, water moves out from the descending lymph into the tissue fluids by osmosis. Thus, a tubular fluid passes down through the descending lymph in osmolarity, that is, concentrations increases gradually due to water loss, and on the other hand progressively decreases due to sodium and chloride secretions as it flows up through the ascending lymph. Besides, whenever water retention is necessary, pituitary secretes the ADH, that is antidiuretic hormone, which makes the cell in the wall of the collecting duct permeable to water. Due to this, water moves from the tubular fluid into the tissue fluid, making the urine concentrated. So here you can see the urine getting concentrated from 300 milliosmol to around 1200 milliosmoles. Students, the cells in the wall of the deeper medullary part of the collecting duct are permeable to urea. As concentrated urine flows through it, urea diffuses from the urine into the tissue fluid and from the tissue fluid into the tubular fluid, flowing through the thin ascending lymph of the Helen's loop. This urea cannot pass out from the tubular fluid, which is flowing through the thick segment of the ascending lymph. DCT and cortical portion of the collecting duct due to impermeability for it in these regions. However, while flowing through collecting duct, water reabsorption is operated under the influence of antidiuretic hormone. Due to this, urea concentration increases in the tubular fluid and same urea again diffuses into the tissue fluid into the deeper medulla region. The same urea is transferred between segments of the renal tubule and tissue fluid of the inner medulla. This is called as the urea recycling, operated for more and more water reabsorption from the tubular fluid and thereby excreting small volumes of concentrated urine. Students, an osmotic gradient is essential in the renal medulla for water reabsorption by countercurrent multiplier system. This osmotic gradient is maintained by vasa erecta by operating the countercurrent exchange system. Here, this is the place where your vasa erecta is going to maintain the countercurrent. Vasa erecta also having a descending and ascending limbs. Blood that enters the descending lymph of the vasa erecta has normal osmolarity of 300 milliosmoles per liter. So here blood that is entering is having the osmolarity of 300 milliosmoles per liter. This is the descending lymph here. As it flows down in the region of the renal medulla, where tissue fluids become increasingly concentrated with sodium and calcium and urea molecules diffusing from the tissue fluid into the blood and water diffusing from the blood into the tissue. So here you can see this diffusion taking place, water diffusing from the blood into the tissues and the ions diffusing from the tissues into the blood. Due to this, blood becomes more concentrated, which now flows through the ascending part of the vasa erecta. So here now concentrated blood is flowing through your vasa erecta. This part runs through such region of the medulla where tissue fluid is less concentrated. Due to this, sodium and the chloride ions and urea molecule diffuse from the blood to the tissue fluid and water from the tissue fluid to blood. So here just the opposite thing is happening. That is, now here again you can see here water from the tissue fluids is diffusing in and the sodium chloride ions along with urea is diffusing out. This mechanism helps to maintain the osmotic gradient. So here an osmotic gradient is maintained which helps for the reabsorption of water. So as to reabsorb water to its maximum capacity, loops of Helen are longer in desert mammal like camels. Due to this, camel excretes concentrated urine. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.